It gives me great pleasure to introduce the presidential lecture. The plan was originally for Caroline Blanchard, which I assume everyone understands is the president of IBNS. Caroline was certainly planning on attending the meeting, but at the very last minute had to cancel due to a family situation, and she sends her sincere apologies. I'll say just a couple quick words about Caroline and then introduce Michael Corley, who's ably going to stand in for Caroline. Caroline, as I said, is obviously the president of the society. She's also a past president of the International Society for Research on Aggression. She's a professor in the Department of Genetics and Molecular Biology at the John A. Burns School of Medicine. Caroline and her husband Robert have published an amazing amount of work in the past 45 to 50 years. They have over 14,000 citations and an H-index of 67. But, as I said, Caroline's unable to attend today. Michael Corley, who is a PhD student in the Blanchard Lab, has agreed to step in and give the presidential lecture. Michael's very capable in doing so. He finishes his PhD in approximately 1 month, so this is almost like a dry run for his PhD defense. Although there's additional work in here, but certainly some of the work that Caroline was going to present is from Michael's thesis. He is a travel award winner as well. He's already secured himself a postdoctoral appointment staying in Hawaii, very fortunate young man. And as it turns out, we believe he will be the youngest ever individual to give the presidential lecture. I give you Michael Corley.
All right. So Carolyn apologized that she couldn't be here today, and unfortunately, she sent her talk to me. And I was going to try to embody Carolyn, but as many of you know, she's a pretty good speaker, so I had to change a couple things. So the plan today is to kind of change her joy of a good model talk into an overview of the Blanchard Lab and what we've been doing in Hawaii in terms of autism research. So I titled the talk "The B2BR Mouse Model." Ethologically relevant behavioral task and the search for mechanisms in autism. And for those of you that have been to Hawaii, the picture in the background is Waikiki Beach. So the lab's 10 minutes away from Waikiki Beach. So, so the overview today, as I said before, the lab's focus is mainly autism. A lot of the previous work in the lab was in aggression, fear, and anxiety. We use basic understanding of those domains when we start to study autism. But the goals of the lab are to understand if behavior is characteristic, autistic individuals can be modeled as precisely as possible in a lab animal. And the second focus, which I'm more involved in, is the search for underlying mechanisms possibly involved in the etiology of autism. So background information, we had quite a few good symposiums on autism in terms of animal models, and then the most recent symposium previously to my talk, basically giving you, I think, a good foundation for autism in terms of what it is. I'll just state simply, when we started modeling autism, I think I got to the lab 5 years ago, and at that time, the lab was starting to get multiple animal models and starting to figure out behaviorally, these models people had created, what was the phenotype like in terms of thinking about ethology and typical animal behavior of the mouse. So when we looked at just the clinical definition on DSM-4, you have social deficits, stereotype behaviors, and communication difficulties. The disorder, as many of you know, is solely defined by behavioral abnormalities. The DSM-5, previously in the talk before, social deficits and stereotype behaviors have been collapsed into one domain, or social deficits, and communication have been collapsed into one domain, and stereotype behaviors of the second domain. But if you're trying to model it, if you can get at communication, stereotypy, and social deficits, you're getting at the same thing. So I don't think it's going to be a big problem in terms of modeling. Just a brief slide in terms of how many are affected. If you look at from 1975 up to currently the most recent CDC report, it's now estimated up to 1 in 50. The methodology for how they get at these numbers is debatable in terms of, is this a true number? But I I don't think it really matters. What we can see is that it's a problem, and we need to start to understand the basic biology of what's going on. So the following slides were in Carolyn's talk, and I think it provides a good foundation for my talk. Why animal models of autism? Some pros and cons she outlined. A pro is the ability to do experimental manipulation. So as many of you work in animal models, you can manipulate systems and single genes. The ability to take invasive measures. So you can take out the brain, take blood samples, and whatnot. Some cons. The animal model obviously can recapitulate the totality of ASD, autism spectrum disorder, and then resistance both, from the outside, and then the emotional in terms of, not wanting to do animal work. So specifically, mouse models of autism, the pros, I think the reason why they've been used are because of the similarity to humans in anatomy, physiology, and genetics. The mouse genome is extensively studied. We have the sequence of multiple strains. JAX has that data widely available. So if you're trying to do, get at underlying mechanisms in terms of genetics, we know the sequence of the mouse genome for multiple strains. There's a wide variety of mice available from JAX in terms of single gene knockouts that you can order up pretty quickly and get in the lab and start to run some of these behavioral tasks, which I'll start talking about in the following slides. Some of the cons, mice are not actually tremendously social species. So my previous work, when I was a master's student, I worked with the rat. And if you were to watch a rat in a cage, you would see that it's quite social and interacts quite dynamically. The mouse, not as much. If you're looking for high throughput, so if you just put the mouse in the cage and observe over a 10-minute period, it will show some indications of sociality, but it's not as dynamic, I think, as the rat. So that's why the move recently has been to rat models, I think. That's why people are starting to go there, just for the behavior complexity that it can pick up. And then this is Carolyn's slide, what she wanted to really, her take-home message, I think, from her talk, was when we're starting to look for the biology of autism, we need to start with behavior. And I quoted her from her talk that she sent over. So behavior is the functional outcome of brain activity. When, as in autism, behaviors are the diagnostic criteria, behavior represents the only clear path to biology. And she also has this concept. She just published a paper. If you look, just type in Carolyn Blanchard, go to her Google Scholar account, on this concept of ethological and functional validity. Cliff Summers is also on the paper. And what she's trying to get at is that if a disorder is defined by deviation from functionality, we ought to have some systematic idea of what functionality is. So animal models should be created on the basis of a detailed understanding of how those same functions are accomplished in the species that has been used as a model. And the best example she gives to me, we start thinking in terms of the mouse is communication. Mice communicate olfactory is their main sense of communication. So, if you're trying to model that, you need to have an understanding of that specific thing in the mouse. And I'm going to give the example of the BTBR mouse when, in terms of an animal model of autism labs been widely working with. And the reason I think Carolyn wanted to focus on this mouse was because it was a nice example of a model that shows a good behavioral phenotype, but also a lot of the data that's generated with this mouse also has the development of novel tasks, which could be utilized for other people's models if you're trying to phenotype your mouse to see, what's the complexity of its autism phenotype and how the mouse acts in tests other than the standardized test. A lot of the work, a lot of the slides I'll be presenting is from Erwin Defenser in the lab. And his whole dissertation was focused on the BTBR mouse and understanding both development and behavior. I'll focus on development or behavior for most of the slides. And for those of you that don't know, the BTBR T plus TF/J mouse model of autism was a mouse that was identified, here, let's go to this slide, in a multi-strain comparison by Dr. Moy et al. Dr. Moy gave a great talk talking about how the three-chamber task, which I'll explain in my next slide, was used to identify the BTBR as showing social deficits. So it's ranked low in sociability in general multi-strain comparisons. It shows in autism-relevant changes in neuroanatomy, immune profile. There's been a lot of publications recently showing the link to autism in terms of mechanisms, but I'll mainly focus on behavior and then give you one indication of mechanism that we're trying to get at in the lab. And if you're looking to learn more about the BTBR mouse, there was a recent review paper from Ksenia Meza in the lab overviewing the general findings of the BTBR. So the goal of Erwin's research was to investigate autism-relevant behaviors of BTBR mice. And when we started in the lab looking at the phenotype of autism models, we start with the 3-chamber to basically establish, is it worth it to do the detailed analyses of the mouse? And a 3-chamber is a social approach task, considered the gold standard for measuring social behavior in the mouse. It's high throughput, so for anybody that runs it, you can basically run a mouse in 20 minutes or so. There's a 10-minute habituation phase and then a second 10-minute test. Typically what it involves, you place the mouse in the center without a mouse in the cup and do a habituation, let it explore freely. And then on the second trial, what you do is introduce an unfamiliar mouse on one side called the social side and put the mouse in the middle and let the mouse explore freely for 10 minutes. And you can record proximity, duration. We mainly start with subject duration to see, does the mouse spend more time in the social side? And this has been, we just replicated previous people's findings in the lab. So if you look at the black 6, which is considered a typical mouse in terms of sociality, what you see is that if you put the black 6 in the center, open the sides, and have an unfamiliar mouse on the social side, the black 6 mouse will spend more time on the social side with the mouse. In comparison, if you do it with the BTBR, you introduce the BTBR in the center, let it freely explore. It doesn't have a preference. It doesn't show more time spent on the social side. And it indicates an impaired social approach preference in the BTBR mouse. Getting at sociality, if you want to make it more comprehensive, what we do, in addition to the 3-chamber, is what Carolyn calls a zoom out and zoom in approach. The zoom out approach I'll explain is the visible burrow system. For those of you that don't know, I'll try to give a good overview of what the test is and how we use it in the lab. The zoom in approach, we call that the social proximity test. And basically, I'll start with the visible burrow system. So it's a semi-natural environment. It's an ethologically relevant test. And what the test is basically trying to mimic is the natural environment of how mice live in the wild. Mice are fossorial creatures. They live in burrows. And we try to take this in the lab and make it as natural as we can. And what we do is we have an apparatus which has 3 living quarters, indicated here, here, and here. Oh, sorry. Here, here, and here. There's a chamber to the surface, so this surface area here is exposed. So this would be equivalent to in the wild when they exit the burrow. And then we interconnect 2 chambers here and have an L-shaped chamber to the outside. The reason there's an L-shaped, the test was initially devised to look at fear and anxiety-related behavior to a cat presented on the surface. So what you'll see is they are able to do like this risk assessment if you have this occlusion here. But we keep the same design and look at sociality. And the way you can get at this is you basically have 3 unfamiliar mice of the same strain. So these mice don't know each other, and you're introducing them into the apparatus, and you look at how they establish a social hierarchy. I just saw a paper right before I got here published in Nature. Someone automated this task, and they called it the Big Brother apparatus. So, you know, I guess that's what they're calling it now. But, and what we do is we look at this for 3 light and dark periods, so over 3 days, and the scoring is time sampled, so we do 6-hour recordings. So you typically start at 6 p.m. at night and then record to midnight, and then start recording at 6 a.m. and then look till noon. And you're able to assess a variety of social and solitary behaviors. So this is what the actual apparatus looks like on the video. And then I'm just presenting 2 behaviors that are pretty simple to understand, but you can look at approach front when 2 mice approach each other from the front, and then you can also look at approach back, which is another indication of sociality, when 1 mouse approaches another to the back. And just a technological thing, the lab has, I think we have 12 of these apparatuses, something like 65 DVD recorders, and a personal blockbuster of behavioral videos, a wide library of videos. So, and when Erwin did this with Roger Pobe, postdoc in the lab, what they saw was that when they did black 6, so 3 unfamiliar black 6 mice, they showed an increase in comparison to BTBRs, which would be introducing 3 BTBRs into the colony, in regards to approach front, approach back, flight, and follow, all social behaviors. signifying that the B2BR mouse was impaired in sociality in the VBS apparatus. And when you looked at the B2BR, what you saw that comparable to other findings that have been widely reported, they do more self-grooming, which may be an indication of stereotypy, which I'll talk about in a subsequent slide. And also the B2BR shows less huddle. Yeah, I can't point over there. So in this apparatus, the B2BR mice showed less social behaviors and more solitary behaviors than black 6 mice. So when you start thinking about the size of your apparatuses, one of the things Erwin got thinking about and the lab widely discussed was size makes a big difference in terms of interaction. If you were to just think about autistic kids, if you were to put them in the room, they'd most likely stay away from each other. VBS is a pretty large apparatus. 3 chambers are a pretty large apparatus. So he started asking the question, what would happen if you confined the space and looked at social proximity and forced interactions? So he's presenting a slide here showing the area comparison of the VBS, 3-chamber, and what I'll talk about now is the social proximity test. And this is our zoom-in test, the social proximity test. And subjects are placed together in a confined space. It eliminates social distance as a variable, and you're able to assess a variety of specific social behaviors and orientations when interaction is hard to avoid. And this is the B2BR mouse in the apparatus. And I'll go over 6 behaviors that we're able to pick up on when we record from this apparatus. Crawl over, so that's when another mouse goes all the way over another mouse. Nose to anal genital. One mouse puts his nose to the anal genital region of another mouse. Nose to face, so one mouse has its nose to the side of the face of another mouse. Crawl under, one mouse goes underneath the other mouse. Nose to nose, exactly what it says, 2 mice put their nose to nose. And then upright, 2 mice rear up together. And when Erwin put 2 black 6, so this is same strain pairs, you're introducing 2 black 6 into the apparatus. What we see is that in comparison to B2BR, when you took 2 B2BR into the apparatus, B2BR mice avoid facial orientations. So they're doing less nose to nose. And then what was interesting was they were doing increased crawl behavior. And what we saw when we started watching the videos was that the B2BR, when it avoided what it would do, it tried to escape and get under the other mouse. So we interpreted this as meaning that the B2BR may be avoiding facial orientations by this behavior. And another thing that was interesting was it showed increased anal-genital contact, possibly signifying it may be searching for a pheromonal cue from the other mouse. But this is something that still needs to be figured out. It has been shown that they have normal sensory olfaction, but in terms of social olfaction, it's still, that field, it's still developing and findings are still coming about. So we don't necessarily know what that really means. And when you introduce mixed pairs, so this would be 1 B2BR and 1 black 6, what you see is the B2BR mice continue to show the increased anal-genital contact, so continuing to search for some olfactory cue from the other mouse. The B2BR mice showed, or crawled under the black 6 mice and remain there, resulting in crawl over by the black 6 mice. And interestingly, B2BR mice did not participate in nose-to-nose behavior. So when the other black 6 mouse, which is typically looking for a nose-to-nose behavior, the B2BR would jerk its head away and not want to do that with the black 6 mouse. And Carolyn thinks that in her talk, what we think the nose-to-nose and head posture signifying the B2BR is maybe a gaze aversion, which is widely reported in autism in young children. One of the things Erwin's working on currently is trying to figure out in younger mice, do they do this? Or even with a mom, if you put a juvenile with a mother, does this nose-to-nose and nose-to-face change? So on to the next, which is repetitive and ritualized or stereotyped motoric behaviors. So being as a part of the Blanchard Lab, we have a great understanding of ethology and basic animal behavior. And one of the things we had a graduate student, Brandon Pearson, was interested in was looking at grooming behavior in terms of structure of grooming to see if this was a stereotyped behavior that could be modeled in animal models of autism. And he went and looked at the just basic understanding of mouse behavior, and Francis Stillwell in the lab of John Fentress analyzed grooming sequences described as correct fixed body area grooming sequences. So typically what you see in a mouse is when they groom, they always usually start at their head and move down their body in a sequence and end usually around the tail area. And he went and looked and discovered a algorithm based on the frequencies of correct and incorrect body area transitions, and used this to see if the BTBR mouse showed some indication of stereotypy. So Brendan looked at the microstructure of grooming in BTBR mice in the Prox apparatus. So the lab's, the underlying principle is basic behavior, and our apparatuses are simplistic in, a sense. This is just a plexiglass, so he put the mouse, introduced it into the plexiglass, and recorded from multiple angles to look at grooming. And what he saw was that the BTBR mouse, in comparison to the Black 6 mouse, showed more grooming, which has been widely reported already by other labs. And it showed an increase interrupted grooming bouts. So it would not continue in the typical head all the way down to the head all the way down to the tail area in its grooming pattern. What it would do would go maybe 1 to 3, stop, maybe 1 to 3 again, stop, 1 to 4. So its sequence of grooming wasn't typical in the sense of how a normal mouse would groom. And what he also did in the same, in his same publication, he wanted to get at some form of cognitive stereotypy. And we had a lab meeting, and one of the things, what we said was, well, what if we used the toy test? Just introduced a novel object to a mouse and looked at what they did with these novel objects. And what you can look at is object approach investigation and the order of object approaches. And you can use this to investigate limited interest and stereotype behaviors in mice. And when you do this with Black 6 and BTBR mice, what we typically see is the BTBR mouse shows more time spent with the toy. So in this case, here's a Lego. So what you'll see is the BTBR mouse will spend more time with the Lego, indicating it has a preference to this toy. And also when you look at its sequence of how it visits toys, it does show a higher incidence of a sequence. So it'll go, for example, the Lego to the jack, Lego to the jack, and then, back and forth, back and forth, back and forth in those sequences when we do a sequential analysis, indicating it does have some form of stereotypy. The Black 6 will usually spend, in general, an equal amount of time at each toy, and then, its sequential analysis isn't stereotyped in that regard. So in summary, the BTBR mouse, for communication and repeated behaviors, seems to show abnormalities, maybe relevant to autism. Others in the lab has also shown reduced scent marking and ultrasonic vocalizations. For those of you that don't know, mice scent mark. So what it is they deposit urine on the ground, and this serves as a form of communication. Typically, you'll see it when you introduce an unfamiliar stimulus mouse. They'll scent mark to basically say, territorial, this is my area. And then ultrasonic vocalization is another form of communication. So mice typically emit like a 50 kilohertz call, which you can pick up with special audio equipment and look at the amount of calling. So what others have reported is that the BTBR mouse, when you put a stimulus mouse, will show reduced scent marking to another mouse, indicating it doesn't really care the other mouse is there. And also its ultrasonic vocalization is decreased, so it won't emit calls to the other mouse. And then I showed you data from the lab indicating increased grooming and stereotype patterns in grooming the BTBR mouse, and then preference for specific novel objects, and then stereotype patterns of visiting novel objects. So to summarize, the BTBR adult male mice display abnormal behaviors relevant, I think, to the 3 core diagnostic symptoms of autism. But when I got in the lab, all of these are adult mice. I was really interested in looking at infants and young. And working with Bob and Caroline, there's not really an excuse to just go on PubMed and do a quick search for behavior. We really have a deep understanding of, basic behavior and papers that go back even to 1953. And what I looked at when I went back and pulled this from the library in Hawaii, there was something called this socialization period by Williams and Scott, and it was from 12 to 25 days. Day 12 being when the eyes of a mouse first start to open. So I got really interested in the question of looking at the development of social behavior and social interaction. And one of the things I had typically heard was we don't typically see play in the mouse, which, I would agree with. I don't see it usually that much. But when you start looking at some of the papers, when you look at wild mice in a dynamic environment, a complex environment, or a natural environment, undisturbed, it is reported they do some indications of this play behavior, which I'll talk about in the data I'll be presenting. So what I did baby VBS, that's what we're going to call this task. And the data I'll be presenting, we're still adding in, so it's in preparation. We used the modified version of the visible burrow system to study litters of dams of black 6 and BTBR mice continuously in the semi-natural environment from postnatal day 12 to 29. And as I said before, postnatal day 12 is around the time black 6 pups' eyes are open and the pups begin to nibble food. So the apparatus, basically, it's just a VBS with just a nest and a burrow out. So the mom is placed in the nest with her pups, and you can look at, when you record, emergence onto the surface. So the surface here is exposed, and then the hopper and food are usually over here. So the data I'll show right now, we looked at 6 litters per strain, and what we did is we looked at the dam with 2 male pups and 2 female pups, so the litters were cold. So we wanted to start just basically looking if we had 2 sibling mate males, 2 sibling mate females, so 4 pups together, and they were weaned at postnatal day 21. And we took 6-hour recordings during the light and dark phase, and we used time sampling scoring. So as I said before, as you could imagine, this took quite a lot of time to score. So this is I think the result of over probably 3 years in the making. But so you have a light and dark for each postnatal day. So I'll start with the location data. So what I've done here is plotted the postnatal day collapsed across, so you're in 3 days. So these are 3 days pre-weaning and 3 days post-weaning. And what you see is the black 6 emerges onto the surface from the chamber rather abruptly during the dark phase, around the time of weaning. So it seems like when you're taking mom away, this is when they start to emerge onto the surface. And this coincides with decreased chamber use. We don't pick up a difference with the BTBR, so they're emerging about the same time. But interestingly, when you look at the light phase, you're leaving them there continuously recording undisturbed. What you see is that post-weaning, the BTBR mouse is on the surface. And what we think this possibly is getting at is an alteration in circadian rhythms, and there was a good symposium on circadian rhythms. We don't necessarily know what this means, but it's a finding I think that really needs to be followed up in terms of looking at the circadian rhythm of this mouse, and specifically all animal models of autism. I think circadian rhythms are a critical component to possibly their social behavior and development. And we also see this in the adult mouse. I didn't talk about that previously, but it is interesting that this is occurring at this early stage of development in the mouse and also in adulthood. And you can see too their chamber use is down. So when they're emerging onto the surface, they're going to obviously spend less time in the chamber. So just to summarize, the black 6 emerges onto the surface rather abruptly, it seems, around weaning. The black 6 settles into low surface use during the light phase after weaning. The BTBR mouse spends more time on the surface during the light phase, both before and especially after weaning, suggesting abnormality in circadian rhythm. And as I said, it supports findings in the VBS of the adult mouse with BTBR mice and in autistic individuals. So when you look at social behaviors, I previously described this approach front, approach back behavior. And what you see is that the BTBR mouse shows, in comparison to the black 6, reduced approach front and approach back behavior following weaning. And this supports findings in the VBS of adult and suggests postnatal day 21 to 29 may be a critical time period for mouse socialization. So this may be the period where they're starting to do these behaviors that are critical for what they're going to show in adulthood. So we looked at crawl over, crawl under. What you'll see if you go back in the literature, this was interpreted when you don't confine the space. Mice that do this at a young age are thought to be play soliciting. So mice would crawl under and over each other as a form of play soliciting. And what we see in the BTBR is it shows less crawl over than the black 6 during the post-weaning phase. However, no differences in crawl under were found. This might make sense possibly why we're not seeing findings. The apparatus is pretty large. So you would expect if you're not confining apparatus, our sensitivity to pick up crawl over, crawl under wouldn't be as great. So, we're still trying to figure out if we can go back in and, rather than time sample, maybe do a more comprehensive analysis to see if there's differences in this specific behavior. And this was one play behavior that we've basically stumbled upon. Bob had some familiarity with it from an early paper in 1971, and what we saw is there was a description of this fast hopping jerking movement with sharp changes in directions in mice at early ages, and they call this a jerk run. So what you'll see is the mouse typically jerks and will run, and when you get this in coordination with two animals, they show an explosive sequence of jerking together and erratic running behavior. They term this popcorn behavior. What I'll do is, let's see, unfortunately, I didn't have a lot of time to prep for the talk, so I wasn't able to get you from the data I've generated, but what I had was the lab tech send over a video, a clip of a quick sequence of jerk and run, and then I think you'll see a popcorn. I think, yeah, so there's a jerk and run there, approach front, and then you'll see one mouse seem to set off the other mice in terms of this popcorn behavior. So when we look at this jerk and run in the black 6 and BTBR mice during development, what we see is that the BTBR mouse displays less jerk and run behavior during the dark phase following weaning. Again, supporting, this maybe mouse socialization period is post-weaning when you take mom away. This is when they really start to interact with each other. The popcorn behavior, we're currently analyzing the hard problem with the popcorn behavior. We don't see a lot of it, so it is hard to pick up on. So, especially if you're trying to make this high throughput, you're probably not going to see it. And then one of the last things I'll talk about is the self-play behavior, so climb and jump. So as you'd expect, the developing mouse probably needs to play with itself in some sense to get an indication of, motor, or to start to develop some motor skills and ability to interact with other mice. And what we do is we look at climb and jump, pretty self-explanatory behaviors. And the BTBR displays less climb and jump behavior during the dark phase following weaning, indicating, another possible abnormality in development in terms of play behavior with itself. So to summarize the results in the social and play activities in the mouse, the black 6 pup displays an emergence of social and play behavior around weaning, postnatal day 21. And social and play activities were much higher in the black 6 control mice compared to BTBR, with these differences mainly seen after weaning. So just to summarize these baby VBS findings, the development of social and play behavior is altered in the BTBR mouse model of autism. And I really think understanding the development of social and play behavior in the mouse is going to be critical to expanding testing of mouse models of autism. And this task, I think, may provide useful for examining molecular mechanisms underlying behavioral intervention strategies in autism. So there's been this, a lot of people talked about behavioral intervention being a form of treatment. Well, if there's some way to socialize the mice with other mice, I know it's been done at adolescence, postnatal day 30 or later, but if you do it at this younger phase and get recovery in behavior and then start to look at the mechanisms in the brain to see, what's underlying these changes in behavior, I think it may be a useful task for doing that. So I'm going to switch gears completely now on the lab search for mechanisms. So I said we're basically working on two sides. On one side, trying to establish good behavioral tasks, and on the other side, trying to understand possibly what's going on in autism in the brain. So we've really been guided by three principles. Where in the brain, so the question, where may we look? And then what in the brain, and then when. I put about asterisks by when, because when you start to do that, we're not at that phase yet in terms of being able to really hone in and do analyses at a young age and compare across development. Yeah, we really need to, a lot of the behavior has been established in adult mice, so I think that's a good starting point, and then we'll get to the when question later. So for where, the lab had worked with the B2BR mouse, and what we saw was that there were a lot of publications in terms of indicating that it showed callosal a thinning of the hippocampal commissure, and a collapsed lateral ventricle. And individuals with autism tend to show thinning or even agenesis of the corpus callosum. And when we started looking at this, we had a collaborator at Hawaii that was looking at a specific region of the brain which was implicated in terms of stem cells and neurodevelopment. And when you look at the mouse, this region's the subventricular zone of the lateral ventricle. And it was first described by Joseph Altman back in the 60s in terms of neurogenesis, that the mouse showed neurogenesis in adulthood. And what you see in the mouse, I'll do a basic overview, is that there's a generation of a proliferation of cells here, and those cells migrate and end up in the olfactory bulb, the region for the mouse that's especially critical, I think, to social behavior and communication. And what we worked with the collaborator on, Dr. Mercier, was looking possibly in this area, why is this area doing this in terms of, what's unique molecularly about this region? And what he had found was that there were unique molecular components of the extracellular matrix. So the environment, the molecular makeup of the environment of the SVZ was shown to have some components that weren't found in other regions of the brain. So we got really interested in this, and one of the things he had found was that there was a specific molecule in this region, heparin sulfate, which was known to sequester growth factors, cytokines, and a variety of other ligands. And what we became very interested in was looking at, was there an alteration in the distribution and magnitude of heparin sulfate in this region in the B2BR mouse? So Brandon Pearson and Ksenia Meza, postdoc, what they did was they started to want to look at this in the mice. And when they went through the literature, what they saw was that manipulating heparin sulfate in the brain was shown to cause callosal agenesis in mice. And then there was a recent study that they came across where heparin sulfate was associated with autism. They did a conditional knockout of heparin sulfate. So if you wipe out heparin sulfate completely in a mouse model, it's not going to work. It's critical to development. The mouse won't be born. But when they conditionally knocked it out, what they saw was that these mice showed an autism-relevant phenotype. So they thought that this possibly was a cause to this autism phenotype in the mouse. So Brandon and Ksenia examined the SVZ region of adult Black 6 and B2BR mice, and what they saw was a reduced lateral ventricle area. And when they looked at the distribution and quantity of heparin sulfate in the region, they saw a decreased quantity and distribution of heparin sulfate in the B2BR mouse, suggesting aberrations in the SVZ niche, the molecular niche, may be involved in autism. So this is where I came to the lab trying to understand behavior, and I was starting to do some translational work, and we started asking the question, do the findings of the B2BR mouse translate to the brains of human autism diagnosed individuals? So just some background information. Why study postmortem tissue for autism? Pros, it's probably the biospecimen containing the biological mechanism involved in the etiology of the disorder. It's a brain disorder, so if you're going to look for a cause, it's probably in the brain tissue of an autistic individual. And it's considered the gold standard in the search for genetic and molecular mechanisms. However, some big cons are very limited availability. These specimens are very precious, and unfortunately, I know there's been a big movement to establish a national brain bank, but unfortunately, for those of you that didn't hear, we did lose quite a few samples. There was a freezer failure at one of the brain banks, and a large portion of the samples, human autism samples, were lost, unfortunately. They're ramping up efforts, I think, to increase the brain bank samples to try to contribute more in terms of what we're, because sample size is a really big problem. And then another con of using postmortem tissue is confounding factors due to life history of the donor. So when I started on the project, I basically asked the question, does the human brain have a SVZ region? Yes, it does, and interestingly, this region is one of two regions in the brain that exhibits neurogenesis in adulthood. And the one unique thing about the human brain is that it has a unique structure in the SVZ in terms of its cellular organization. So what you see I'll give you some basic anatomy. Lateral ventricle, we should probably know, everyone knows where that is. The subventricular zone lies next to the caudate, so it's basically on each side of the lateral ventricle. And when you look with immunofluorescence at the organization with DAPI labeling, what you see is that there's an ependymal layer, and then there's this gap. There's no cells in this gap, and then there's this astrocytic ribbon, and this was shown by Nader Sinai and Alfredo Quinones' group. So I just want to establish the structure, just some of the sides from the data you'll see. Well, this will make it a little clearer. So when you look at structure again, there's an ependymal layer, a hypocellular gap, which is devoid of cells largely, an astrocytic ribbon, which is full of astrocytes, and then what they call this transitional zone. This is where you start to see neurons appearing, so this is probably into the caudate area. And then, as you can see, some of these astrocytic processes do extend through the hypocellular layer into the ependymal wall. And when you look at the SVZ in the human brain, one of the things I was thinking when I got started in this, well, it projects to the olfactory bulb. The olfactory bulb is not very critical to the human brain, but interestingly, the human brain shows two migratory streams. You see one to the olfactory bulb, and then there's a medial migratory stream that is in the infant human brain, and this contributes significantly to the prefrontal cortex, which we know is widely implicated in autism in terms of disturbances. So this got me really excited in terms of looking at this region in terms of its potential to possibly be involved in some of these growth abnormalities that we see in the autistic brain. So why focus on the SVZ region? I gave you some rationale. Let me just hit the point a little bit further. It's a niche for neural stem cells during prenatal brain development. It coordinates stem cell proliferation, differentiation, and migration. It's one of two sites in the brain, human brain, for neurogenesis and adulthood, hippocampus being the other, which has been widely studied. And it's modulated by the environment, which is another interesting factor. And then just to link it back to autism, it's been proposed by a lot of people that have looked at the neuropathology of autism that there's this dysregulated brain growth throughout neurodevelopment, specifically Eric Corchain's group. And what they see is an overgrowth early in development, and then this arrested growth later in development, which even sometimes is an undergrowth, so the brain seems to decrease in size. And what they proposed was that the findings strongly suggest looking the mechanisms they said was cell proliferation, cell death, cell migration. All these things hit the SVZ region, so I think these findings strongly suggest looking at the SVZ region in the human brain. So what we did was we asked three basic questions. Can we locate the SVZ region in postpartum tissue samples? Does the molecular microenvironment of the SVZ region differ in the autistic brain? And then one of the last things was just a follow-up experiment. What is the cellular proliferation activity in the autistic brain? And what we did was we obtained postpartum tissue through the Autism Tissue Speaks program. To start, we chose 5 individuals. One of the samples, unfortunately, wasn't of adequate quality to be cryosectioned, so it had to be omitted. But we got 4 cases ranging from age 5 to 60 of individuals diagnosed with autism, ADIR diagnosed, and we obtained matched controls, matched based on postpartum interval and sex, so all these are males. And I think just an interesting thing I'll highlight, when you look at the 5-year-old in comparison to the typically developing, you look at the neuropathology report, you can see 1560 grams, 1320 grams overgrowth, which has been reported. And then by 20 years of age, this is the typically developing, 1440 grams, 1144, so this arrest in growth. And then I won't spend too much time on the ADIR, but it's been a good tool for postpartum tissue in terms of getting diagnosis after an individual dies to confirm that this is an autism diagnosed case. But I'm just presenting the ADIR scores here in the 4 domains of the diagnostic interview that's administered. So all cases met autism diagnosis. And then the workflow, so what we did, the Brain Bank, what they do is they obtain the whole fresh brain from the donor, and then they separate the brain into two hemispheres. Left hemisphere is fresh frozen typically, and then the right hemisphere is formalin fixed. They do this because the formalin fixed side could be used for structural analyses if you want to do like electron microscopy or look at structure, and then the fresh frozen side is done so you can start to do some molecular analyses. And we're focused on the molecular side, so we obtained frozen coronal sections. We're interested in the SVZ region, so as you can imagine, we did 3 tissue chunks, and we really were looking, you want the corpus callosum on the top and the caudate on the side, and we're interested in this region here. So we obtained 3 tissue chunks from each individual, mainly towards the anterior portion of the lateral ventricle, because this region has been shown previously to be thought to have the most proliferation activity. So just methods of the study design, we cross-sectioned these tissue chunks, immunolabeled, did confocal microscopy for image capture, and then used ImageJ for image analyses. And what we see is that we were able to locate the SVZ in autism diagnosed in typically developing brains. So as I said before, the typical structure, what you see when you label with DAPI, I'm not presenting the GFAP labeling here. I didn't think it would come up with the green. But lateral ventricles here, you see that ependymal layer 1, layer 2 here, the hypocellular layer devoid of cells, layer 3, the astrocytic ribbon, so this would be if you label with GFAB, where you'd see the astrocytic ribbon, and then 4 is the transitional zone into where you start to see some neurons. And we asked the question, we started, well, what would we see if we labeled for heparan sulfate, what we saw in the BTBR mouse? And what we came across was that the localization of heparan sulfate, interestingly, was more in the hypocellular layer in comparison to layers 3 and 4, just in the typically developing. And when you looked at the autistic brain, what we saw is the youngest individuals' samples showed a reduction, drastic reduction in the distribution and quantity of heparan sulfate in comparison to their matched control, suggesting there's an abnormality in this molecule in terms of the extracellular matrix of the SVZ region. And we got thinking that, well, okay, we know that heparan sulfate is thought to bind growth factors. Let's see if we can measure cell proliferation in the SVZ region. You know, you obviously can't inject BRDU in a postmortem individual, but what you can do is use Ki67, which is a marker for cell division. And you wouldn't expect to see much in terms of dividing cells, but interestingly, what we saw was when we looked at the 5-year-old autism diagnosed individual, it showed more proliferation in comparison to the control, suggesting maybe there's this abnormality in cell proliferation in this region that's still there, post-mortem, but, you know, it's suggestive, I think, at best in terms of, you know, getting at what's actually causing it. The experiment we're performing now, which is trying to look at, we're labeling for a variety of growth factors. It's just been problematic in terms of the correct antibodies and specificity to the human brain. So just to quickly summarize the immunohistochemistry results we found, there's abnormal quantity and distribution of heparan sulfate in the SVZ region of the youngest autistic brains in comparison to typically developing brains. There's indications of increased proliferation in the youngest autism brain in the SVZ region, so I think this is interesting. Unfortunately, we're not able to look earlier in development, specifically because diagnosis doesn't occur usually before 3 years of age, so, you know, it would be really hard to look prenatally at cell proliferation in this region to get at some of these questions. And then we think aberrant extracellular matrix function in the SVZ brain region may contribute to dysregulated brain growth in the young autistic brain and potentially be involved in the etiology of the disorder. So if you're getting this overproliferation of cells, I'm pretty sure stuff isn't getting to where it needs to be correctly. And, you know, there's going to be all these downstream effects from this. And then the translational aspect, the BTBR mouse showed a similar thing. We're trying to work that out in terms of manipulate the system to see if it affects behavior and then get at some of these developmental questions. But as a start, I think it's a pretty good biomarker translation in terms of looking at the mouse system and going to the human system. And then, so I still have 15 minutes. So I'll switch gears again and just try to tell you what I'm working on currently. So none of this, I'm prepping this data right now for publication, but I think really this is where the real, you know, the major results and breakthroughs I think are going to come from other labs, and I hope, you know, possibly from our lab, the Blanchard lab as well. So I'm really interested not just looking at single molecules and single systems. I'm interested in looking at networks of things and how networks of things change. So the project I'm currently on right now and what I'm working on is looking at molecular and epigenetic analyses of the SV region in autistic postmortem brain tissue. And the goal of my research is really to identify molecular and epigenetic aberrations in this region in autism diagnosed individuals. So the approach I've been taking, just to give you some history of gene expression, I'm really interested in looking at which genes are turned on and off, but not using a simple, fast, single gene method. I'm really interested in using modern technology, and one of the rapid advancements that's occurred and has been proposed is, a big breakthrough in terms of how we look at some things is next generation sequencing. And specifically, when you can do that, what you can look at is you can look at gene expression by taking messenger RNA, converting it to cDNA, and then mapping that to the human genome to get an indication of which genes are turned on and off. And this approach is, I think, novel because it's unbiased in terms of we don't have to make a probe. We have the human genome sequenced, and what you're doing is looking unbiased at gene expression and mapping. And what you can see is, genes we know and what they're involved in, but also stuff we don't know. And I think this will really be a major breakthrough. So what I did, I have the tissue sections, so you're able to isolate RNA from these tissue sections. I've increased sample size. I won't get into the increased cases that I've added to the study, but so you isolate RNA. You need to test for RNA quality. RNA quality is a big problem for postmortem tissue, as you can expect. Once someone dies, RNA probably starts to degrade, but we do see good quality RNA. Typically, our RNA integrity numbers are usually about around 7 or higher for most of our samples, so we've been pretty good at getting high quality RNA. And then there's library preparation, so you just need to take the RNA, convert it, and make a library that you can sequence. And then sequencing occurs, and bioinformatics analyses. I won't go through RNA-seq. I don't have time, but just one interesting fact. All the sequencing I've been doing occurs on Coconut Island. So for those of you that have been to Oahu and know Gilligan's Island, it's on the opening scene of Gilligan's Island. And seriously, we have a sequencing machine, Illumina, on an island, and I have to take a boat to get the sequences sampled. The reason this occurs is because they got money to sequence the coral. As you know, global warming, they've been doing a lot of sequencing work on coral. But yeah, we have a sequencer, and it can be used for other things. So that's what I've been working with. And then I won't go through how you quantify gene expression. What I'm most interested in, so these results just fresh, being prepped for publication. But what we see when we look at the youngest age comparison, so we compared young autistic samples age 5 to 8 to match controls age 5 to 8, and we grouped gene differences. Typically, we see about 1,000 genes are changing. And I've been working with a bioinformaticist to look at, what does this necessarily mean? And what you see is when you look at molecular and cellular functions, when you group, we see changes in cell death and survival in 259 molecules, cell growth and proliferation, cellular development and differentiation of cells, cellular movement, migration of cells, tissue morphology, quantity of cells. And these findings are in support of previous studies that have been done by Eric Corchain's group, and then Daniel Geschwin at UCLA has also shown some good work with postmortem tissue. So I think we're really building a good case in terms of, this growth abnormality and mechanisms. But the thing I'm, really excited about is the ability to look at 269 molecules and how they change together and cluster in terms of a network and start to dissect out these questions. And the last thing we did was we went to, the bioinformaticist went to a database of genes that have been associated with autism at some level, and we get a hit rate of about 7.5% of genes that have been associated with idiopathic autism, and then when you look at syndromic autism, known single genes, 6.1%. And then the last thing I'll just talk about is we also did this and looked at the oldest age group, so these are individuals from 30 to 60 years of age. We don't see as dynamic a changes in terms of gene expression, maybe somewhere in the order of 300 to 500 genes changing. But what we do see is what's changing is different. Molecular transport, neurotransmission, long-term potentiation are changing. So suggesting there's this dynamic change in gene expression in terms of autism. So if you're starting to look at it, each individual is different, I think, but the other problem is age. Age is a real big factor in terms of, when you're doing discovery and trying to think. So this is, something we're trying to work out. I think, the benefit of what I've been doing is this data can be deposited in a repository and available to everyone. So, as more people start to do it, we can start to take everyone's data together and start to analyze, reanalyze stuff, because the data is deposited raw. So, the bioinformatics side, we can go in, reanalyze as new cases are added, and then even look at regions and start to do this by region. So, I'll have data on the SVZ region and other people prefrontal cortex, so you can start to look at the interplay of multiple regions. Skip through this stuff. So just to conclude, the BTBR mouse model I talked about shows an autism-like behavioral phenotype, capturing the 3 core symptoms in standardized and ethologically relevant tests. We still need to figure out why the BTBR mouse does this. The mouse wasn't created. Nobody knocked a gene out of the mouse. It's inbred strains, so we still need to figure out why this is happening. I told you about heparin sulfate, but I think it's more dynamic than just heparin sulfate. There's other growth and guidance factors and a variety of other things that are changing the brain that may be involved. And then autism may reflect widespread changes in brain hydrology and function, resulting from gene by environment based alterations in growth and guidance patterns. I think originating in the SVZ region. This is a lot of people have proposed growth, cell division, and this hypothesis, I think this is the heart of where stuff is going wrong. And the future plans, I think what I'm going to build my career on is to utilize my understanding of animal models to start to demonstrate some of these gene by environment changes while continuing to analyze the human tissue, because I think that's really going to drive a lot of the work in terms of the animal side, in terms of what we target. So just to conclude, acknowledgments, the Blanchard Lab, and then I just really want to talk about the Autism Tissue Program, and I think it's a vital resource in terms of autism research to really provide to researchers to start to contribute to autism research, and then specifically the families of donors, individuals that have a kid that unfortunately has an accident and they're donating their child's brain. I think is to science is one of the utmost things you could do, and then just NIMH for grant support, and then Bob and Carolyn just wanted me to tell everyone that they sad they can't be here, but IBNS has been a critical component to their career, and they hope everyone has a good time in Ireland. So that's it, and I'll open up for questions. Hopefully I can answer some stuff. If not, you can email Carolyn. So.
Thank you very much, Michael. That was quite an impressive talk, and certainly suggests you've got a very bright future. We have essentially used up our time, but we'll take 1 or 2 questions if there are.
So I was interested in specifically some of your work, which showed that some of the behavioral trajectories for the BTBR mouse seems to be normal up to the point of weaning. Yeah. And I wondered, can you think of a way that you might be able to postpone weaning or artificially supplement that period to see whether BTBR mice trajectory following that normal period of weaning would look more normal, or whether it would diverge at that point anyway?
Yeah, so that's, that was an interesting thing in terms of when we first designed the study, was when we were going to wean. Because a lot of people even wean further than 21 days. We just chose 21 as a nice middle ground in terms of when we can wean. I think the mother's behavior isn't different. I didn't present the data, so that was a big question. Is the BTBR mom a bad mom? Is this why they're not doing this? So that's not different. But yeah, I think the next experiment, it's going to take another 3 years maybe to do it. But to manipulate, to move the weaning period to see if that has altered. Or even, wean them earlier to see, at the earliest stage, and then see maybe you get a recovery when you do that. Because, a lot of labs are on standardized weaning days. They just wean on the same day every time, and I think you're right. That makes a big difference in terms of development, what's happening. So it's, obviously a great thing to consider when you're designing a study. So good question. Thanks.
You almost showed us the mean of the behavior of your autistic mice. But I suggest that there are also like 6 mice which show some autistic-like behavior, and there were also some of this autistic mice which show normal behavior. Did you look also into the brain of such mice, and what do you see, for example, in the subventricular zone?
Yeah, so the data I presented, we did collapse across. We did identify each mouse, so we have each mouse identified, and then we have basically, what that individual mouse has done. We did take blood samples and brain we just need the funding. I really want to get the brain sequence. That's what I would really want to do. Specifically, I want to look at the SVZ region in the BTBR and Black 6 mouse at these different points and see. So yeah, we did save the samples. So luckily, we'll have a way to coordinate the behavior. It's after, though, post-node day 30. So, we don't get to see on that day if they're impaired on 21. We didn't sack mice at 21. We sacked them at the end of the experiment. So, but yeah, we have the tissue samples, so we can start to look at that.
Okay, I think we've used up our allotted time, but please join me in congratulating Michael once again.